So picture this. The human genome is like a book, a massive book that contains the entire blueprint for a human being. Now, we spent decades, just reading every single letter in that book. But just knowing the letters, well, that's not enough, is it? We have to understand the words, the sentences, the story. So how do scientists go from just reading the code to actually understanding what it all means? Let's dive in. Take a look at this number, 3.2 billion. That's how many letters, or nucleotides, are in our nuclear genome. We've got the whole sequence written down, which is, an absolutely monumental achievement. But as you can imagine, it also presents an even bigger challenge.
Right, and that's the multi-billion-dollar question, isn't it? We have the sequence, but what does it all do? What's the function of all these genes? And what about those huge stretches of DNA in between them? Answering this is the whole point of a field called functional genomics. To solve this mystery, scientists have had to become, detectives. You can think of the genome as our crime scene, and every single gene we don't understand is a person of interest. Our mission? Figure out exactly what role each one plays in the incredibly complex story of life. Okay, so what's the first step in pretty much any modern investigation? You run a background check, right? Well, in genomics, that means we head straight to the computer. We check our massive databases for any known associates or, any prior records. This first step is called a homology search, and the logic behind it is actually pretty simple. If our mystery gene sequence looks a whole lot like a gene we already know, let's say a gene from a mouse that helps build muscle, we can make a pretty good guess that our gene has a similar job. It's kind of like finding out your suspect's entire family are carpenters. It's a really strong lead. But here's the catch. This digital work has its limits. Sometimes genes that look related have actually evolved to do totally different things. Other times, the gene we match it to is, it's also a mystery. And every now and then, we find no match at all. So the trail goes cold. The bottom line is, we can't solve this case just by sitting behind a desk. So, when that digital trail goes cold, it's time to get our hands dirty in the lab. It's time to bring our suspect, the gene, in for some serious questioning. We've got to move from just predicting to doing some direct experiments. This is a classic detective move, right? If you really want to know how important someone is to an operation, you just see what falls apart when they're not around. In genetics, we do the exact same thing. We systematically turn a gene off, or inactivate it, just to see what happens. Now, the most famous method for this is creating something called a knockout mouse. It's a really complex process, but it's incredibly powerful. Scientists engineer these mice so they're completely missing one specific gene. By studying what goes wrong with these mice, what processes fail when that gene is gone, we get direct, hard evidence of its function. This whole approach really gives us two main strategies. On one side, you have loss of function. That's like removing a gear from a machine to see what stops working, like the knockout mouse. Then, on the other side, you have gain of function, where we basically make that gear spin totally out of control to see what happens. Both of these tactics give us absolutely critical clues. Okay, so up to this point, we've been interrogating the master blueprint, the DNA. But what if we could actually listen in on the real-time communications happening inside the cell? Instead of just reading the static code, what if we could basically tap the phone lines and monitor the active messages being sent out? Well, these messages are made of RNA. The main tool for this kind of cellular surveillance is the microarray. The best way to think about it is like a giant switchboard that lights up for every single gene that's currently on and sending out messages. It gives us this incredible snapshot of what a cell is doing at a very specific moment in time. And this is where things get really, really powerful. By comparing the gene activity fingerprint of a healthy cell to that of, say, a cancer cell, we can pinpoint exactly which genes have gone rogue. We can literally see which ones are shouting when they should be quiet, or which ones have gone silent when they should be active. Our investigation is getting deeper. We've identified our suspect, we've seen how it behaves, but here's the thing. Genes, just like criminals, almost never work alone. So the final piece of this puzzle is to figure out their network of accomplices, the other proteins they're interacting with. This is the key question if we want to understand the bigger picture. A gene's function is so often defined by the other proteins it works with to get a job done in the cell. We absolutely have to map out this social network. To map this network, scientists use this incredibly clever method called the Yeast-2 hybrid system. Think of it like setting a molecular trap. Our mystery protein is the bait.
Then we test thousands of other proteins as the prey. Now, if a prey protein sticks to our bait, it triggers an alarm bell that goes off in the cell, and bam, we found a direct connection. It is a brilliant way to systematically find all of a gene's accomplices. So you might be thinking, does all this detective work actually work? Does it solve cases? Oh, absolutely. Let's look at a real-world example of how these exact techniques were used to crack one of the very first major genomic mysteries, the genome of Baker's yeast. Okay, let's rewind to 1996. When the yeast genome was first sequenced, scientists found 6,274 potential genes. That's a huge number for such a tiny little organism. But here's the really wild part. For 2/3 of those genes, that's over 4,000 of them, scientists had absolutely no idea what they did. The vast majority of the yeast genome was a complete and total black box. So what did they do? Well, they did exactly what we've been talking about. As this quote explains, they launched a massive, systematic project. They went through gene by gene, knocking them out one by one, and then carefully watching to see what went wrong. It was a colossal, painstaking feat of genetic detective work. And this, right here, is why it all matters. Every single gene we can assign a function to, even in a simple little organism like yeast, is a new clue to understanding our own biology. This is how we find the roots of diseases like cancer, or Alzheimer's, or diabetes. This kind of detective work is truly the fundamental basis of modern medicine. You know, the incredible thing is, this investigation is far from over. We still have thousands of our own genes whose functions are either a complete mystery or are just poorly understood. Each one of those is like a locked door. So the real question is, as we keep developing these amazing detective tools, what profound secrets about our health, about aging, about life itself are just waiting for us to unlock them?